Hi, everyone, and welcome to today's seminar. It is my pleasure to introduce our speaker for today, Dr. Thakur. Shalu is a postdoc in the Vegetable Breeding and Genetics Lab. She's been with us since January of last year. And our project is basically looking to develop novel germplasm for disease resistance in cucurbits, so squashes and watermelon. Before joining TREC, Shalu worked as an independent fellow at the Indian Institute of Pulse Research. And then she did a PhD at the National Institute of Plant Biotechnology and a master's. So for PhD, she did on fungal blasts in rice, developing breeding tools for disease resistance. And then for master's, she did a work at the Agricultural and Forestry Institute University and a Bachelor of Science from HPKV, I believe. So with that, our talk today will be on genome editing in cucurbits, current status and future prospects.
Thank you, Dr. Meadows. A very good afternoon to everyone. So I welcome you all for my presentation. Today I'll talk about genome editing in cucurbits, the current status and future prospects. So first, it's not moving. So this is the outline of my talk. I will talk about the discovery of CRISPR-Cas9, then how this technology, the CRISPR-Cas9, has been developed into a tool, a genome editing tool. What are the application of CRISPR-Cas9 and opportunity in cucurbits? So first, what is gene editing? As name suggests, gene editing means to edit particular gene or genome. Gene editing is also known as DNA editing technology, genome editing technology, and CRISPR-Cas technology. So how this gene editing technology works? It works like a biological version of word processing program, which use find and replace command. When this guide RNA and DNA complex, we deliver this complex into the cell. This guide RNA look for the complementary sequence in the genome. Wherever this guide RNA found the complementary sequence in the genome, the Cas9 cut the DNA at the specific location and then make the desired changes. So what is the difference between genetically modified organism and gene-edited products? I'll talk about technical differences. The genetically modified organism, a foreign genetic material has been inserted into the genome, and resulting organism has the genome with inserted foreign piece of genetic material, 1 or 2 pieces of genetic material, or any new gene has been inserted, but from different organism. Whereas in GE product, no genetic material or no foreign material has been introduced into the genome. Only the existing genome has been edited or modified. Now, the second is the natural occurrence, likely or unlikely. The genetic modified organism, it is highly unlikely. We cannot find genetically modified organism in the nature. It can be created in the laboratory through specific technology, which is genetic engineering technology. Whereas GE product, the synthetic product which is developed in the lab can be similar to the product which is occur in nature, or we can create it through conventional breeding methods. So now look at the regulatory guidelines. The genetically modified organism, these are heavily regulated. So biosafety guidelines to release these products are in place. And genetically modified product, when it reaches to market, it will reach in market around about 14 years. It will take 14 years, a time frame to reach the genetic modified organism or plants into the market. Whereas GE product, the regulation is a bit relaxed. So the product will reach like less than 5 years into the market. It is easy, simpler and easy to create the genetic edited product as compared to genetically modified organisms. So this is the map showing how genetically edited crops are regulated across the globe. So the countries which are indicated in green, here, these particular countries, the gene-edited products, the regulations are very relaxed. Whereas European Union, New Zealand, these countries, the gene-edited product is regulated as GMO. They are treated as GMO, whereas few countries like Norway, there is no guideline for the gene-edited product. So if we have to develop the plant variety using any of the technology available, how much time it will take place to develop a variety? So with the help of crossbreeding, it will take like 8 to 10 years to release a variety. Similar with the mutation breeding again take 8 to 10 years to develop a new variety, whereas transgenic breeding take a bit of longer time, more than 12 years. And with gene editing technology, it is possible to develop a variety in less than 6 years. So that is the power of the gene editing technology. The gene editing technology using CRISPR-Cas9 is a very powerful technology to develop a new plant varieties. So gene editing technology I mentioned earlier also, it is a CRISPR-Cas9 technology. So what is basically CRISPR-Cas9? CRISPR-Cas9 is an acquired immunity in the bacteria. Bacteria has a different type of immunities. First is the innate immunity, which is the natural immunity present in the bacteria, and another one is the acquired. Acquired is a CRISPR, which bacteria acquired with the time. So innate immunity, innate immunity has different defense systems, which is inside the bacteria, which include absorption block, injection block, replication block, RM system, and cell suicide. So when bacteria sense that there is an attack of bacteriophage on the bacteria, so they try to activate absorption block. So they don't let bacteria to sit on the surface. They don't let bacteriophage to sit on the surface of the bacteria. If bacteriophage manage to sit on the surface, then at the second stage, the second line of defense is the injection block. In this defense, the bacteria don't let bacteriophage DNA to inject into the cytoplasm. But if they surpass this defense also, the third line of the defense in the bacteria is a replication block. So once the bacterial DNA is injected into the cytoplasm, it starts replicating. And once it starts replicating, the bacteria has RM system. So they have some restriction endonucleases, which try to cut the foreign DNA which has entered in the cytoplasm. If the bacteriophage manage to surpass all these defense, 1, 2, 3, and 4, the last defense is the cell suicide. So bacteria, what they did, if they don't manage to kill the bacteria, they kill themselves. So cell die. Then another immunity which has bacteria developed over the time is the adaptive immunity, and this is known as the CRISPR. So let's know in detail what is CRISPR and how it works in bacteria. So CRISPR is basically an acquired immunity in bacteria, and it stands for clustered, regularly interspaced, short palindromic repeats. So palindromic repeats are the sequence which you can read like similar in both the direction. If you read from 5'3" or 3', right? It will be the same. For example, racer car. Racer car, if you read from left to right, it is a racer car. And again, from right to left is a racer car. So CRISPR stands from cluster regularly interspaced palindromic repeats, and how CRISPR locus look in the bacteria. So this is the architecture of the CRISPR locus in the bacteria, which include Cas9 genes here. And then they have some repeat sequences. And in between the repeat sequence, they have the spacer sequences. This whole locus is known as a CRISPR locus. Here, point. Okay. So this whole this is the architecture of the CRISPR, and this whole locus is known as a CRISPR locus. So the spacer, this is the footprint of the bacteriophage. For example, when this bacteriophages infect the bacteria, so bacteria took a part of the DNA and integrated into its genome. When these bacteriophages attack, then again bacteria take a part of it and put as a spacer in the CRISPR array. So this spacer is the genetic material of the bacteriophages, which they integrate over the time in the CRISPR array. We also visualize this CRISPR array is a vaccination card of the bacteria. So with the help of this spacer sequence, we can know like how many bacteriophages bacteria has encountered throughout the life. And this spacer sequences is very important for the bacteria for acquired immunity. So now let's see how this CRISPR array works to protect the bacteria from the attack of bacteriophages. So this is like when the bacteriophage attack the bacterial cell, this is an acquired immunity. When they inject their DNA into the cytoplasm, Cas1 and Cas2 enzyme, they cut the piece of the bacteriophage's DNA very precisely and very accurate nucleotide base pair. And after cutting a very precise sequence, which is known as spacer, they integrate this spacer into the CRISPR array, which is adjacent to this repeat motif. And when this CRISPR array, the new spacer has been acquired between these two repeat elements. And after that, during the transcription process, it synthesizes into pre-CRISPR RNA. There is a trans RNA molecule already present in the bacteria, which has a complementary sequence to this repeat element. Now, at this time, this trans RNA, it binds to the complementary sequence to the repeat element. And then at the same time, Cas9 also bind to this spacer trans RNA and cRNA complex. Now, once this complex is formed, RNase3 cleave this complex into an individual complex, which is known as a surveillance complex. So this is a surveillance complex. So every surveillance complex, we also known as effector complex. They have a CRISPR RNA, trans RNA, and Cas9 protein. So spacer RNA is a sequence which is complementary to the sequence of the bacteriophage. So for example, this bacteriophage has infected the bacteria, and bacteria has acquired a part of the DNA into the spacer sequence. When these bacteriophages again try to reinfect the bacteria, so this surveillance system is already all the time active in the bacterial cell. It starts looking for the complementary sequence in the bacteriophage DNA, and the moment they found the complementary sequence in the target genome, they just bind there and Cas9 cut it and just break the DNA, and no infection takes place. So that is how CRISPR-Cas9 acquired immunity works in the bacteria. Sorry. Till now, many CRISPR-Cas9 acquired immunity has been described and identified, and they have classified into different classes, class 1 and class 2. CRISPR-Cas9 is a class 2 type 2 acquired immunity. For class 1, CRISPR-Cas class 1 type of the Cas9, they need multiple Cas9 for target cleavage and the binding, whereas type class 2 Cas9 proteins, they need only single Cas9 to do the particular job. So these Cas9, CPF1 and C2C2, these Cas9 protein are multi-domain protein. So they have a several domain which have several functions. So how Cas9 interact with guide RNA? Before 2012, the Cas9 structure has not been identified. Dodona group at the same time, University of Tokyo has crystallized the Cas9 structure, and they found that Cas9 is 1369 amino acid long and 163 kilo, the molecular weight is 163 kilodalton. This is how inactive Cas9 complex look like. They have basically two lobes. One is the target identifying lobe, which is also known as the PAM interacting, and another is a nuclease lobe. Nuclease lobe has two domains, HNH domain and RUBC domain. With the help of these two domains, the Cas9 binds to the two strands of the DNA. And when this Cas9 combined with the guide RNA, they form active complex, which is also known as a ribonucleocomplex or effector complex. And this complex then start looking for the target sequences and then cleave the target DNA. So this is the timeline of the CRISPR discovery. In 1987, a few repeat elements has been identified in the E. coli. But by that time, their role in the immunity has not known. So later on, one another group of researchers has identified similar kind of repeats in archaebacteria. And then in 2005, how these repeats, the role of CRISPR loci has been established. And in the same year, 2005, the Cas9 protein and their function has been reported. So 2007, CRISPR RNA has been discovered. Till this time, there was the one missing puzzle, which has not. So these till 2007, they don't know exactly how this CRISPR-Cas9 complex work in the adaptive immunity. With the discovery of tracer RNA in 11 and 12, this missing piece has been discovered, and they exactly know how this whole system works, and how they impart the immunity to the bacterial causes. So this CRISPR-Cas9 in 2012, with the discovery of this tracer RNA, hijacked into a genome editing tool. And in 2015, in the Science journal, they have named CRISPR as Breakthrough Technology of the Year. And after that, the CRISPR is unstoppable. The researchers all over the globe, they have started using this technology and started manipulating the genome according to their wish. So 2020, the Charpentier and Jennifer Doudna, they awarded the Nobel Prize in Chemistry for the work, for the discovery, for the development of the method for gene editing that allows rewriting of the code of life. So these two ladies, they have developed the CRISPR-Cas system into a programmable gene editing tool, which we are using nowadays. Before 2011, these two women scientists, they don't know each other. So interestingly, they went to Puerto Rico in a meeting, and where this Charpentier at the time was working on pathogenic bacteria, mainly on the Streptococcus pyogenes gene, how this bacteria cause infection, and Doudna, most of her work is on the RNA, which is a cousin sister of the DNA. So after the meeting, they went to the cafe and they started discussing about their research. And then they started, at that time, that talk led to the discovery of the CRISPR-Cas9 system. So they started thinking, like, if the CRISPR-Cas9 creates the break in the bacterial system, can we reprogram it into something something like genome editing tool, which can make the precise cuts in the DNA? And after the meeting, within a year, they have published the breakthrough CRISPR-Cas9 editing technology in Science in 2012. So the CRISPR is a very powerful technology. With the help of CRISPR, you can make any changes, the desired changes where you want in the genome. So this gene editing technology using CRISPR-Cas has transformed all the editing technology. Before we have few technologies like zinc finger nuclease, talent, but the none of these technology has as powerful as the CRISPR. So with the help of the CRISPR, it is easy nowadays to design the crop, any crop for better nutritional crop, high yielding crop, disease resistant crop to feed the growing populations. Now comes to the IP issues. In 2012, the Dodna group, they have filed for the patent. And in the same time, in December, the Broad Institute, Dr. Zhang was also working on the CRISPR. He was working on editing the human cells, and he also filed for the patent. And in 2014, the patent has been granted to the Broad Institute. At that time, Dodna group was little disheartened, and then they started appealing and re-appealing, and a few rounds of the courts. In 2019, both the parties got the equal patent right. So few companies, they got the patent from the Broad Institute, and few companies, they got the patent from the Harvard Institute. But UF patent firm, in 2012, they again filed the case, and up now the days, the patent is with the Broad Institute with the Dr. Zhang group. But at the same time, the Jennifer and Carpenter has a patent right in some of the European countries and some Australian countries, but they don't have the US patent. Only Broad Institute, the US patent. Now, US patent is with the Broad Institute with the Dr. Zhang group. So now, summing up the journey of the CRISPR-Cas from nature to the lab. So in nature, the CRISPR-Cas has three components. It is a three-component system. Bacteria, they need CRISPR RNA, tracer RNA, and along with the Cas9 to cleave the particular DNA. But Dodna and Charpenter, they have used a linker, and these two components, they have joined together to a single component, which is easy to synthesize in the lab. And this is correspond to the target sequence, which we want to change. And now this modified system, which work as gene editing tool, has two molecule complex, which is sgRNA and Cas9, and we also known as effector complex. The Cas9 is a programmable enzyme, we also known as a molecular Caesar. We can program it the way we want, and which uses different sequence of RNA to recognize the particular DNA or the gene of interest. So the Cas9, it cuts the DNA. Cas9 cut the DNA, it acts as a molecular scissors. I already mentioned many times about the Cas9, it acts as a molecular scissor, it creates a double-stranded breaks. So once the DNA cut, how it gets repaired? Every cell has a repair mechanism, which is known as non-homologous end joining repair mechanism. So when Cas9 creates, when this complex, guide RNA spacer, this is known as a single gRNA complex. With the Cas9, it forms an effector complex. When these complex, the spacer sequence, it is user-defined, and it is correspond to the target sequence. When it binds to the target sequence, this complex break the DNA into double-stranded. So at this time, by non-homologous end joining repair mechanism, this has been repaired, and at the site of repair, insertion, deletion, or frame mutation take place. That is how editing happens. The another repair mechanism is homology directed repair. This is similar to the NHEJ, but the only difference, a template which we want to insert here, we supplied with these, the template has been supplied from the outside. So these are the few successful examples of gene-edited crops which are available in the market. The first one is the guava tomato. The guava tomato has been discovered by Xanthii Seed. This is a seed company based in Japan with the name of Ceylon Rough. And this particular tomato is rich in the GABA. So they have manipulated this GABA shunt pathway, and because GABA synthesis is high in the GABA synthesis take place from flowering to mature when the tomato is green in color. But once the tomatoes start ripening, the GABA content start decline. So they have manipulated this pathway and they have developed a variety which is rich in GABA. And this variety is already available in the market, in the Japan. Another gene-edited product where they have increased the vitamin D content in the tomato by modifying a duplicate section of the phytosterol biosynthesis pathway. In the rice, the broad spectrum resistance edited line has been developed by creating the mutation in the promoter of sweet 13 gene, and the resultant line, they impart robust and broad spectrum resistant against all the bacterial blight or Pseudomonas oryzae species. In wheat, they have targeted the scientists have targeted the MLO locus. They knocked down this locus and developed the edited line, which has more resistance as compared to the wild type. So now, I already talked about what is gene editing. Gene editing has been adopted as a technology, but how we can use this technology in our research, how we can design our experiment for gene editing. So gene editing technology basically relies on these 5 factors. The first, accurate identification and the characterization of the target. Then repurposing of the molecular agent. What are the molecular agents? The plasmid, the Cas9, and the HRNA guide. Then how to deliver, once the reagent is ready, how to deliver this reagent inside the plants. And once it is successfully delivered, how to regenerate that edited line. And once this edited line has been developed into plants, so how to screen the edited line for the editing. Let's talk in detail one by one. The first is trait identification and characterization. So to start with any gene editing work, it is very important to identify the trait which you want to work on, and that should be well characterized. How we can characterize it. For example, if I want to change the color of a flower, so first I will look the gene which is responsible for color of that particular, for example, red color. So I will look for the genes which are responsible for the red color. If there is no report in the literature in that particular flower, and nobody has reported or worked on that particular gene, then I will start looking in the literature for the related species. If the gene is available in the literature species, in the related species, I will take the sequence, design the primer, and try to amplify that particular gene in the genotype I am interested to work. Once I get the sequence, I'll try to translate the gene, try to study their expression, when the gene is expressing, how much is expressing, at what stage it's expressing. After getting all this information, along with this, I would like to check on the allelic variants in the gene pool. If different flowers, that flower color, and in that particular species, they have another flowers which have different color, I will amplify that particular gene in all the contrasting phenotypes, and then I will try to look upon the variation in that particular gene pool. So first, I need to show about the trait, like the trait I'm working is the flower color. Then at the same time, I should be very sure about the gene which is responsible for that particular trait. So once I'm sure and I have the sequence of that particular gene, next step is to design the guide RNA. So for designing guide RNA, there are a variety of softwares are available. You can design, you can retrieve that sequence, or you have the sequence of your gene. Just put the sequence in the software like CRISPR, ChopChop, CTOP, and then you can design the HGRNA based on your requirement. At this point, it's very important to know like which promoter you want your guide RNA to drive. If we are working in the monocode, we need different promoter. If we're working in dicode, we need different promoters. And then what kind of Cas protein we want for my experiment. Is Cas9 will do the work which I'm planning to do, or I need the Cas12. They are a little bit different from one another because there are a lot of Cas9 variants available in the database. Basically, if you're Cas9, they recognize NGG promoter, and there are other variants also available. They have different recognition specificity. So once I decide upon the reagent, my HGRNA, Cas9, HGRNA is driven by which promoter, Cas9 is driven with which promoter. One, I'm sure about HGRNA and Cas9, and then I clone these into the vector. After cloning, this is the biggest hurdle. How to deliver that particular reagent into the system. For plants, the most commonly used is agrobacterium and particle gun method. But for most of the crop species, the tissue culture, it is very difficult to regenerate and transform. And the transformation efficiency is also very less in few crops. And editing was also not that efficient. So the delivery of the reagent using these two systems is the biggest hurdle. So we need to look for some alternate approaches, which is regeneration independent or tissue culture independent. After once, with the help of agrobacterium mediated transformation or genetic particle gun transformation, I transferred or I delivered my construct into the seeds or the tissues or the explants. Then the second step is the regeneration of the epiphyte. How to regenerate the epiphyte? Which explant has maximum regeneration potential? Sometimes we are using, for my experiment, I have used a variety of the explants starting from hypocotyl, cotyledons, then axillary, cotyledonary node, axillary meristem, explant. None of the explant works in the tissue culture. None of the explant regenerate into the plantlet except for the cotyledonary node explant. So I'm using in all my experiment the cotyledonary explant. So it is very important to standardize the regeneration and the transformation protocol for the crop which you are planning to work. So once you are able to regenerate with that particular construct, the plants, then the next hurdle is to identify the edited lines which have no foreign DNA. So first step is to identify the negative segregant in the progeny. We can isolate the DNA, and when we can design the primer specific to the construct, and we can look for the negative segregants. We can go with the whole genome sequencing. Then once we eliminate these two negative segregants, then the positive segregants, we need to look for off-target identification. If like the off-target means the space of the sequence, the single guide RNA, they edit the target sequence. Along with the target sequence, they edit somewhere else in the genome or not. So Off-target identification is very important. And the last one is, once we are sure that the plants, they have only editing and they don't have any off-target editing. So then the next step is to maintain that with the backcrossing. This is the work I have done in India. I was working in India as an independent research, and my work was funded by DST Inspire. So there was an BS1 gene, which is known as big seed 1. The time I started work on this particular gene, so the function of this gene is not known in chickpea. In soybean, they have proved through RNAi that this gene is responsible to increase the seed size. So I started looking for the homologs in chickpea. I retrieved the sequence, tried to look for the allelic variants in the database. We have a contrasting chickpea genotype available in my institute, having small size, big size, medium size. So I want to check the function of the gene. Is BS1 gene has a similar function as they have in soybean? So when I got the edited lines, and then this is a control, and this the edited line. When I compare the phenotypic characters of the control and edited line, so all the edited lines, they have increased in leaf size, stem length, port size, and seed weight has also been increased in the transgenic line. So this BS1 gene in the chickpea also has the same function as in the soybean. So now what I'm doing in cucurbits, in cucurbits, there are few reports. The people has worked to edit few of the locus in cucumber, watermelon, and xanthii has modified the erecta gene in melon, squash, and cucumber. So the technology is constantly evolving for more precision, and this technology has immense potential. So when, so in the squashes, in the lab, we don't have any transformation, regeneration protocol on the melon, on the squash. I'm working in the squash. So I tried to disrupt PDS gene. Why PDS gene? Because its phenotype is easy to visible. You don't need to go for the genotype. Just when you bombard, when you design the HGRNA specific to PDS gene, you regenerate the cell and you can see the phenotype. So this is a proof of concept in this cache. So I started looking for the PDS gene in the genome, and I found 2 homologs in the cucurbit database. And then I designed the HGRNA for both the homologs. These 3 modules has been designed. So the first module having PDS, one HGRNA from the PDS1 gene, another HGRNA from the PDS2 gene. Another module has both the HGRNA from the PDS1 gene, and the third module I have designed, which have both the HGRNA of PDS2 gene. So HGRNA has been designed using CRISPR software. The off-target has been checked in off-finder, and the HGRNA with a minimum off-target or no off-target has been selected. And finally, PHSC1 vector has been procured from the ad gene. The vector, I received the vector in the form of staph culture. I maintain the glycerol stock. I confirm the vector with the restriction digestion PCR. Once the vector has been constructed, at the same time, I have synthesized the HGRNA, and then I try to assemble these HGRNA using cloning method in this particular vector. So this is the right border to left border of this vector. So they have these 2 HGRNA. This HGRNA1, it is driven by U6 promoter. HGRNA2 again with the U6 promoter. Cas9 has been driven with 35S promoter. And there is a plant selection marker, hygromycin, which is used to select the transformants. Once this construct is ready, it is the reagents for genome editing. So once this construct is ready, then I send this sequence for this construct for sequencing to confirm the cloning. So for transformation or for delivery, I have used particle bombardment system. These are the squash explants. So seeds, the squash seeds were washed with water, surface sterilized, and put in the media, and then let it grow for a week. After a week, explants were prepared. This is called latenary nodal explant. And when the explant was ready, this construct was coated on the gold particle and delivered with this ballistic machine to the explants. And then after the several rounds of subculturing on shoot induction media, we see the albino or bleached phenotype. From albino and bleached phenotype, I have isolated the DNA and checked for the integration of tDNA. This tDNA is the part of this complex. The primer has been designed from here to here to amplify in the putative transgenic line, and the lines which amplify these particular bands has been sent for the sequencing to check for the editing at the targeted site, and at the targeted site, single base pair deletion 2, or in some transformant 3 base pair deletion has been observed. So the transformation efficiency has been achieved is 4%, which is very less. And we know that the cucurbits crops are very hard to transform and regenerate. So the efficiency is very less for the cucurbits. So this study has opened up the opportunity to study the gene function for basic research and trait improvement in the squash. So we know that the powdery mildew is a devastating disease of cucurbits. It infect the plants at all growth stages and reduce the marketable yield. No resistant gene has been identified till date against the powdery mildew in the commercial available variety. Of course, there is a resistant gene available in the wild, and the resistant gene mediated, our gene mediated resistance is race specific, and it can overcome easily as the pathogen is constantly evolving over the time. When the pathogen is evolved, the R mediated resistance will be break down. So what is the alternate approach? How to tackle this powdery mildew disease in the squash? So we try to disrupt the susceptibility gene. And by disrupting susceptibility gene, we'll be able to get the broad spectrum and race non-specific resistance. We'll look at the watermelon genome and the squash genome, and we found 14 homologous in the watermelon and 18 in the Thakur. We try to construct the phylogeny of all the MLOs which is available in the database, in dicots and the monocots. In the cluster 5, it is already reported in the literature that the MLO locus which clustered in the 5 is a candidate for the susceptibility. So these 3 genes, one of watermelon and these 2 of squash, they cluster together in the cluster 5, and in one of the researchers, they checked these genes against the powdery mildew infection, and they found that this is upregulated. So then I decided on, let's start work. Let's target first these 3 genes. So now the next step is to design the sgRNA. So one approach is to design the separate sgRNA for every gene, and then make a 3 construct, and then deliver this construct into the plants. Another approach, which is a little time saving, is just look at the sequence of all the 3 genes. Multiple alignment has been done, and I looked for the conserved sequences. So I designed only single sgRNA, which can target all the 3 genes together. And a single construct has been prepared. With a single construct, 2 sgRNA, 3 genes have been targeted in 2 different watermelon and the species. So for transformation, I'm using the genotype Black Beauty and early prolific. The transformation protocol and regeneration protocol has already standardized in the Black Beauty. A little bit in EP, but in watermelon, Mikili, and sugar baby, we have already standardized the transformation protocol for these 2 also. So these 4 genotype has been currently I'm using these 4 genotype to, with the single construct, which is targeting all these 3 genes together. So this is once the construct is ready. This is agrobacterium mediated transformation. Once we prepare the explant, the agrobacterium carrying that particular construct, we pick a single cell from the media, from the plate, and culture it overnight and let the culture grow. When the OD reaches 0.6 to 0.8, then go with the secondary culture. Again, put this culture, the 5 ml of culture in 50 ml, let it grow for more 24 hours. Again, measure the OD. When the OD reaches 0.6 to 0.8, pellet the agrobacterium. After pellet, dissolve it in the half MS media and infect your prepared explant with the agrobacterium. These are few of the methods to increase the efficiency, which is already mentioned in the literature, and then try to regenerate the plants through that particular in agrobacterium infected plants. One of the problem which we face basically in the tissue culture with agrobacterium transformation, agrobacterium most of the time overgrow in the plates. So that is the biggest hurdle, but still sometimes we get few explants or few regenerated plants. So this is the few plants which I'm able to get after agrobacterium mediated transformation. I haven't checked here for the editing. So till now, what are the gene edited products are approved and available in the market? The first one is the conscious green. Pure Wise has developed this product. Then the cultivated wheat, Sicilian rough, I already said, and the white button, which should. So here to sum up my talk, the CRISPR-Cas9 is a curiosity-driven technology. It is, if we know the target, if we know the gene, it is very, if we know the target, we know the gene, it is very easy to develop the crop with high yield, nutrition on the quality, better nutritional quality and disease resistant. Because this technology is very efficient, very powerful, and very simple to use, and very precise. But the basic problem is in the cucurbits is the transformation and the regeneration, because the transformation and regeneration efficiency after the delivery of the reagent is very less. So for increase the efficiency in the cucurbit, we need to optimize the reagent for efficient editing. We need to change the reagent, or we need to develop some genotype independent regeneration and transformation protocol, which can be used in any crop, any genotypes. So here I would like to thank my PI, Dr. Meru. So I'm very thankful to you, Dr. Meru, for giving me an opportunity to work in your lab. So he's always motivating and inspiring me. My lab mates, Prerna, Swati, Eugene, they are always helping and very cordial in the lab. I'm very thankful to you all. So the funding for this project has been received from FDACS, and I'm thankful for all the people in Tropical Research Center. These are my institute, Indian Institute of Pulses Research, where I worked on genome editing and I learned the technology. My institute, from where I graduated, and Department of Science and Technology and Inspired, from where I get the funding to work on genome editing. So at last, I would sum up with the words of the Norman Borlaug: everything else can wait, agriculture can. Thank you so much.